we said that from here to here, right, from jagged line to jagged line, was this repeating unit, like boxcars on the myofibril train. What's it called? Yeah, it's called sarcomere, exactly. And then what we did was we simplified it, and instead of showing it 3D, which is what it is, we showed it 2D, right, so that you could actually sort of look at the structures of the sarcomere. And then what we were doing this time was saying, okay, we're going to look at the structure of a sarcomere, and we're going to say, okay, Z line to Z line, also called Z discs. That's a sarcomere. And inside the sarcomere, there's basically this pattern of proteins. And there's two proteins I really wanted you to know about. These two are the ones that are primarily involved in contractions. There are these thin proteins that extend to the Z lines, and then there are these thick proteins that are in the middle. And we started talking about the contractile process, but before we get deep into that today, let's talk about how this structure gives you striations again. And then we'll talk about contraction, and then we'll talk about how the striations change upon contraction. Ready? Okay, so if I used a protein staining technique and I had thicker, denser proteins, then of course it would be darker in that location, right? So what we get is we get dark wherever we have myosin, right? Does that make sense? Because myosin's a thicker protein. So what's going to happen is this part right here, wherever I have myosin, right here, is going to be the darkest band. It's called the A band. Remember it because it's the second letter of dark, D-A-R-K. What it stands for is anisotropic, which you don't give a crap about, do you? And isotropic is I, but it's also the second letter of light, okay? So I have this A band, right, wherever I have the myosin, right? And then I have this I band wherever I don't have myosin. Does that make sense? So basically that will correlate to what we looked at last time, which was this, right? So if we look at skeletal muscle up really close, we'll see an A band, dark, and then an I band, and then an A band, an I band, an A band, an I band, right? Okay, this is a different staining and a different type of microscopy. This one shows it in more detail, and you can see other stuff that you can't see with that type of microscopy. Okay, so we get the A band and the I band. Notice that in the middle of the A band, if you've got really good staining, you can see that it's actually slightly lighter in the middle. That's called the H zone. Okay, so now what I want you to understand is what is going to happen to that banding pattern when you contract the muscle. Here's a relaxed muscle. What we said before was that muscle contraction or sarcomere contraction works like this. When stimulated, the muscle, the myosin heads are going to reach up and grab active sites on the actin, yeah? And then they are going to pull. And then what happens to the distance between the Z lines? Shortens, okay. But concept check for you. Does the myosin change length? No. Does the actin change length? No. What they do is slide past one another, yes? And they are filaments. So they call this the sliding filament theory of muscle contraction. Does that make sense? The myosin and the actin filaments slide past one another, the sliding filament theory. Okay, so now what I want you to do is try to conceptualize how that is going to change the banding pattern. When I took physiology back in college, which was like 25 years ago or something, we actually still killed animals in physiology just to see what was going to happen. And I know that sounds terrible, but don't think that I forgot the lessons when I killed the damn frog, because it didn't. Okay, so we anesthetized the frog, and in order to understand this, what we actually did is we took his gastric medius, and you know what that is, and we stretched it out over a light microscope, and then I zapped it with an electrode to watch what happened in the banding pattern of the muscle when I stimulated it. And what happened is, whereas relaxed muscle is nice and striped, what do you predict about contracted muscle? What's it going to be? It's going to be dark. It's going to be almost completely dark. So what happens is this is relaxed. So look here. Look at the length of the I band for a second, right? This is partially contracted, yeah? This is completely contracted, yeah? And then look at the H zone, right? And that's that lighter portion right in the middle of the A band. Wide, narrow, gone, right? So you could zap it and you could see it go from striped to dark, striped to dark, striped to dark, right? That's part of how they figured out what was going on with the sliding filaments, is they just had this experimental indication that they thought the filaments were sliding past one another, And then later on, when we got different protein staining techniques and we could stain actin and myosin different colors, you can actually confirm that that's what's going on. That makes sense? So it's called the sliding filament theory of muscle contraction. It is the basis both of the striations and also of the mechanics of muscle contraction. Note to self, it seems kind of complicated now. This is nothing compared to how detailed you'll learn it in physiology. So you learn it at this level now, and then you'll learn it way more detailed. You have to add in a bunch of other proteins and wherever the ATP is used and all of that kind of stuff. Does that make sense? Okay, so A-band, I-band, H-zone. So the basis is basically the sliding filament theory. When you stimulate with a certain type of neuron, called a somatic motor neuron, you'll learn that later, the actin and the myosin slide past one another and the Z lines come closer together. The H-zone shortens and the I-band shortens. Does the A-band shorten? No, the A-band stays stationary, right? It pulls everything into it, right? So the filaments don't shorten, but the banding patterns do, right? And the distance between the Z lines does, okay? All right, so now what I want to do is I want to go gross anatomy all the way down to submicroscopic and then back up so that you guys can figure out how these things are related to one another. Does that make sense? Okay, so you ready? All right, so first is, let's go back to the gross anatomy for just a second. And let's say, okay, we're talking about gross anatomy. We're going to go all the way down to submicroscopic to help you guys understand the relationships of these things to each other. Okay, an entire skeletal muscle, the whole thing, is wrapped in connective tissue called what? What's the connective tissue? The epimysium. Good. Okay, and is composed. The whole skeletal muscle is composed of multiple bundles of muscle fibers. What's a bundle of muscle fibers called? A fascicle. And each individual fascicle is wrapped in connective tissue called what? Perimysium, yes. And is composed of multiple muscle cells, or what's the other word for a muscle cell? Muscle fiber, okay. Each muscle fiber is wrapped in connective tissue called what? Endomysiums, yeah. Deep to the endomysium, you run into the muscle cell membrane, also called the what? Sarcolemma, yes. Deep to the sarcolemma with all of the other organelles and stuff, you have a bunch of cable-like bundles of contractile proteins, okay? What is one of those bundles called? Myofibril. It's attached to the cytoskeleton. Break the myofibril down, and we can say, okay, if the myofibril is the train, the boxcars, the repeating units along the myofibril are called what? Sarcomeres. Break the sarcomere down. The thicker protein of the sarcomere that stays stationary upon contraction is called a what? Myosin. And the thinner one that moves upon contraction is called actin. Got it? That makes sense? Okay. So let's do it all the way back up. Ready? Okay. So now we are going to start building muscle contraction. I'm going to ask it slightly different way. The band, the A band that is primarily dark, is composed of which of the two contractile proteins? No, myosin. It's mostly myosin. Now there's a little area where actin overlaps it. The area where you don't have myosin is going to be lighter and composed primarily of what protein? Actin. Okay. The repeating pattern of actin and myosin from Z line to Z line is called a what? Sarcomere. Add up one sarcomere after the other, after the other, after the other, you get a cable-like bundle of proteins attached to the cytoskeleton called a what? Myofibril. Okay. Wrap the myofibril with all of the other organelles up into a cell. Okay. The whole cell is called a what? Muscle fiber. Okay. And it is wrapped not in connective tissue. First, give me the cell membrane name. Not the connective tissue yet. Sarcolemma. Okay. Superficial to the sarcolemma, you run into the first connective tissue called what? Endomysium. Now take a whole bunch of those cells, wrap them up together into a bundle. The bundle is called what? Fascicle, right? Whole fascicle is wrapped in connective tissue called what? Perimysium. Take all of those fascicles with their perimysium, wrap them all up, make a muscle out of it, and it is wrapped in connective tissue called what? Epimysium. Epiperiendo. Right? Okay. So if you can do it all the way from macro to micro, then it's one story, right? Not two entirely separate stories, and really there's no trick questions when you can do it all the way from macro to micro. Everybody good? Okay, so the last part on this set of notes is simply to compare and contrast cardiac muscle with skeletal muscle and smooth muscle with skeletal muscle. We're going to learn cardiac muscle in more detail when we get to the heart chapter, but for right now we're just going to look at it in comparison to skeletal. Okay, ready? So cardiac muscle, not surprisingly, has some similarities with skeletal muscle, and the primary similarity is, of course, that it's striated too. So does it surprise you at all that it is striated because it has the same basic arrangement of actin and myosin, and therefore the same banding pattern, the same contraction mechanism? Yeah? So it contracts very similarly to skeletal muscle. You don't stimulate it the same way, right? Like with skeletal muscle, I told you that each individual cell needs to be individually stimulated or else it's paralyzed. Cardiac muscle, that's not true, right? So can't you stimulate this guy right here and it'll tell its neighbor? Yeah? Why? What do cardiac muscle cells have between each other? Gap junctions. So they can communicate to one another, so you don't have to individually innervate every single one. In addition, although we'll talk about it in more detail later, cardiac muscle also has the capacity to do spontaneous contractions, right? I don't know if you guys have ever seen if you can like take a heart out, it'll actually still beat when it's like disassociated from the nervous system as long as you dump the right ions, and it doesn't last for very long, but it can, right? Can still do that. Okay, so it's striated. It doesn't need to be individually innervated from the nervous system. When it is innervated, it's always involuntary. Now with skeletal muscles, you can control them voluntarily most of the time, not all the time. But with cardiac muscle, you can't control it voluntarily. What I'm meaning is if your heart rate is going crazy, can you just use your cerebral cortex, your conscious mind, to slow it down? You actually can't. And I know that you're thinking, well, what about people who meditate to slow down your heart rate? What you are actually doing is you're using your conscious mind to override the part of your brain that is actually in control of your heart. And it ain't very efficient, is it? Right? Doesn't happen instantaneously, doesn't even happen dependably for most of us. But what I'm saying is the voluntary or conscious portion of your brain is not even wired to your heart, right? That makes sense? So it's involuntary in its activity. As far as the basic anatomy goes, the fibers are shorter and there's one nucleus per fiber instead of multiple. And you have those thick parts where the cells come together are called intercalated discs, and they basically look like a really dark striation. What they are is the site where you have both gap junctions and then what kind of cell junctions keep them close, stuck to each other. Desmosomes. And since those are proteins and you use protein staining, they get really dark right there. That makes sense? Okay, so that's cardiac muscle. You go into it in more detail when we get to the cardiovascular system. And then the last one is just smooth muscle. Of course, smooth muscle is called smooth because it's not striated, but which would kind of imply to you that it doesn't have the same sarcomere pattern, but it actually is pretty similar because look here for just a second. Okay, this was a skeletal muscle sarcomere, right? Okay, now look at what smooth muscle has inside. Do you see that this little critter looks pretty much like a sarcomere? I mean, it doesn't end in a Z line, but it's got actin and myosin, right? So why do you not get striping if you have actin and myosin? Because the reason you get striping here is because all of the myosin lines up, right? But here, they're running at all different directions, and so you don't get any striated pattern. Is there still a similar contraction mechanism in which the myosin reaches up and grabs the actin and pulls? There is. It's a similar contraction mechanism. It doesn't end in Z lines, it ends in these little dense bodies. And since they're going in all different directions, when you pull, you don't just shorten in length, you also kind of squish. You blob it up. See that? Right? Which is fine. It's good for the wall of a hollow organ like the stomach. You don't just want it shorter, you also kind of want to squish it, right? So that's how smooth muscle is similar versus different. So the cells are much smaller. They're like eyeball shaped or spindle shaped. It's non-striated. It's also involuntary. You can't decide it's an inopportune time to have diarrhea and make it stop, right? It's really never an opportune time to have diarrhea. And then as far as the functional anatomy of the smooth muscle, what is going to happen is, generally, there are exceptions to this. This picture isn't in your textbook, but It's from a different textbook. Generally, when you've got smooth muscle in the walls of all of your tubes and all of your hollow organs except your heart, which has cardiac muscle. And so how do you get things to move through tubes? What you usually have is usually 2 layers of smooth muscle. So watch. Here is a hollow organ, like maybe a small intestine. You're doing a cross section. And when you look at the small intestine, what you'll see is usually 2 layers. So imagine this is my small intestine. The layer that is closest to the lumen is called the circular layer, and the cells go this direction. So stick with me. When I contract those cells, what's going to happen to the diameter of the lumen? It's going to decrease, right? You're going to squish it, right? And then along the outside, the more superficial layer of muscle is going to be a longitudinal layer, right? And the longitudinal layer is going to go this direction. And when you contract that layer, what happens to the length of the tube? It shortens, right? So here is what you do. You kind of use these muscles as antagonists to one another. And if you're trying to move something through your digestive tract, what you do is you squish it while you're relaxing the longitudinal layer. So it'll become narrow but longer, yeah? And then you pull the longitudinal layer while you're relaxing the inner layer, right? So you squish and then pull. Squish and then pull, right? And as long as you start it from the proximal end and go to the distal end, then you don't get poop in your esophagus, which is good, generally, right? And part of the way that you make that happen is this peristaltic wave of contraction occurs because the cells in smooth muscle have gap junctions in them. So if I start the contraction up here, it'll go squish all the way down, yeah? And then pull all the way down. Does that make sense? And you just alternate the squishing and pulling, and you can move stuff through peristalsis. You can see that really well if you've got your digestive tract stretched out like in a snake. Have you guys ever seen a snake eat? Snake will eat a whole whatever amount, right? And then you can see it squish and pull back, and then squish and pull back, and then squish and pull back, all the while taking the nutrients out, right? And you get a mouse loaf at the other end that's smaller than the mouse that came in that end. That makes sense? So it's peristaltic contraction, and that's what goes on. And it doesn't just go on with solids. Interestingly, why would you need peristalsis to go on in your ureters that lead from your kidney to your urinary bladder? Any idea? Yeah, because we continue to make urine even when we're laying down asleep at night, right? So even to move fluids, right, we're like, oh, you don't need it because of gravity. Well, you're still going to move it even if you're standing on your head. So you do need peristaltic contraction in the walls of those tubes. Is that good? Okay, so that's smooth muscle. Not all smooth muscle has gap junctions, but the one in your intestines does. Okay, and then the next thing that we are going to move to is skin. Okay, and so I know you guys did skin in lab a while ago, but you don't do it in much detail in lab. So this will allow us to finish skin before your lab practical, and then we'll probably finish it on Thursday. And you will have a quiz on Thursday, but let's talk before we get into skin about what your quiz is going to be. Your quiz on Thursday is going to be muscle, all the way down to the microscopic level. But how am I going to ask you about muscles that you're learning in lab? This first quiz on muscle, because you have another one next week to help you get ready for your lab practical. The first quiz on muscle is going to be muscle logic and muscle mechanics, more than muscle memorization. Does that make sense? So for instance, I will tell you the attachment points of a muscle and then ask you the action. I will say, this muscle is attached to the humerus on the anterior surface, and it's attached to the radius on the anterior surface, and I'll give you some choices for actions. Some of them will be at the shoulder, and some of you guys will pick that, right? But what did that muscle do? Blank at the blank. Flexion at the elbow, exactly. So I'll do questions like that over muscles you're actually learning to reinforce the muscles that you're learning, yes? And then I will also do the other kind of question, which is, this is a muscle's action. Which one of these is the origin and which one is the intrusion? Does that make sense? Okay? So this is going to be, do you understand muscles? And then the next quiz that you'll have next week will be, are you ready for the memorization and the understanding at the same time, right? So this one will be like, can you figure it out? And then the other one will be, did you actually memorize all the stuff you're going to know? Does that make sense? So on Thursday, first thing, we'll have that muscle quiz, and it'll also have some of the microscopic muscle structures on it. Good? Okay. And then we're going to do skin. I don't know if we'll quiz on skin or not. We'll see if it goes on that next one next week, but it won't be on this first one. Okay? All right. So integumentary system, which is skin. You did it a while ago. The reason I didn't do it when you were doing it in lab is because you needed my help with muscle more than you needed my help with skin. So we'll just go back and pick it up. And then after this, we'll start nervous system, which will be the next thing you guys do in lab too. Okay? Go ahead. Oh, yeah, go ahead. Yes. Because this time it's just logic, right? That makes sense? This one is going to be appendicular and axial. I don't think I have very many axial on there, but I have some because it's not memorization. It's just like if you put it here and here, what would it do? Or if it did this, it would be attached here and here. Okay? Next week, you guys will all be done with both axial and appendicular, and then I can ask you memorization questions too. Next week would be, here is a specific attachment point. This muscle is attached here and here. Which muscle is it? Right? Like, what's its name? Not what does it do. That makes sense? That takes a little longer for you guys to absorb and pull back up. So we're going to do it in stages to try to like help you get the stuff down. Okay. Integumentary system is chapter 5. It's not very hard. It's not very long. Okay. I will try to make it interesting because there's some culturally interesting stuff about skin, of course. So there's two different ways to approach looking at the skin. You can approach it as a body system, or you can approach it as an organ. So why could it be either? The reason the skin can be an organ is because it's two tissues, right, functioning together. The keratinized, you guys should know because it was on your test, right? Keratinized stratified squamous epithelium, and then a combination of denso-irregular and areolar underneath. Okay, so that's why it's an organ. Why is it an organ system? Because that's not all that's in there. Look at all of those glands and blood vessels and nerves that are in there, even some muscles that are in there. That's why it can be considered a body system, okay? When you look at it as just an organ, though, it's the biggest organ in your body. Not considering subcutaneous fat. Depending on your body size, it weighs between 6 and 9 pounds if you stretched it out. Okay, I know it's gross to think about stretching out human skin, but you know people have done it, so we're going to talk about it. Okay, so 6 and 9 pounds, and it varies pretty dramatically in thickness. So you guys tell me. It varies from about 1.5 millimeters to about 4 millimeters. Now, I know you know where it's thickest, but do you have any idea where your skin is thinnest? Any idea? Yeah, eyes, first places to get wrinkles. Right around your eyes, right around your lips. They also dry out really easily. So, and then where is it thickest? Yeah, the feet and palms, especially in Utah, because it's dry as hell here, right? You'll get hooves if you don't take care of them, right? Have you noticed that? Didn't happen in Texas. I got here, I was like, apparently I have hooves, right? And I'm like, yeah. And it's composed of 2 layers, right? We have the epidermis and the dermis, and we'll talk about those today. The epidermis is here. The dermis, even if the skin is thick, the dermis is way thicker than the epidermis. And then this area down here is not part of the skin proper. It's called the hypodermis of the subcutaneous layer, but it's always taught with the skin, okay? But the skin is only 2 layers, okay? And this layer down here is, of course, mostly areolar connective tissue, and then depending on where you look in your body and how much subcutaneous fat you have, that's going to be adipose as well. And we'll talk about that after, even though it's not part of the skin. Okay, so what kind of stuff is in the dermis? I don't do very many dermis structures for testing. I just don't. I don't know why. There's other stuff I want to spend time on, but there's a bunch of stuff in there. There is erector pili muscles, right? There's innervation in there. There's sweat glands and there's oil glands. So sweat glands, I do want you to know the general name of a sweat gland. What's the general name of all sweat glands? Sudoriferous. And the general name of all oil glands? Sebaceous, right? And then in lab, you learn some subtypes of those, okay? Logic question for you. Sweat glands and oil glands, endocrine or exocrine? Yeah, those are exocrine, Okay? All right. So let's start talking about functions of the skin. We don't spend a lot of time thinking about the functions of our skin at all. But in order to get an appreciation for what your skin does for you, I want you to think about this. How many ways would you be at risk? What kind of ways would you be at risk if you, for instance, had lost your skin protection by having third-degree burns over a decent amount of your body? Does anybody know any ways in which you are at risk if you lose that intact skin protection? Infection is huge. What else? Dehydration, absolutely. You can't thermoregulate, right? So I don't know if you've ever been in a burn ward. They are hot. They are so hot because you have to keep them relatively close to body temperature. So dehydration, all of infection. The other thing, so probably doesn't make any sense to you unless you're in physiology or have taken physiology. The other thing that happens is when you damage all of these cell membranes, your ions that were inside your cells dump into your extracellular fluid, and it actually runs a really, really high risk of heart attack. So all kinds of things are problematic when you damage intact skin. So let's see what it actually does for you when it's intact. It is, of course, protective, but it's protective in ways that you think of and protective in ways that you don't. I know this sounds kind of dumb, but the fact that it's actually got dead skin on the surface means that anytime I rub up against something, I'm not actually damaging living cells, right? So just the fact that it's keratinized on the surface protects you from mechanical abrasion. And then it also is protection from microbial invasion, any kind of pathogens getting in, because intact skin and the big layer of keratin that you have on the surface is relatively impervious to most pathogens. Now, there are flesh-eating bacteria, but thank goodness they don't exist more frequently than they do. Most of the time, in order to get an infection, you have to have broken skin, right? So it's this physical barrier. In addition, your skin also secretes an acidic secretion. So if you lift your skin, don't do it, it's awkward. If you lift your skin, it's salty, but it's also tangy. And the tang is because the secretions on the surface of the skin are slightly acidic, which makes it inhospitable to many, many bacteria. So it's doing both active and passive protection. And then in addition, your skin, if something gets in there, it also has immune cells, white blood cells in the skin. Most of them are in the dermis, but there are a few in the epidermis that keep things from actually going deeper in. In addition, the white blood cells in your skin also protect you from yourself, because you get abnormally dividing precancerous cells all the time, and as long as your immune system is otherwise healthy, you are going to catch most of them, just not all of them. You'll catch most of them. Okay, so the other thing that it protects you from is UV damage, and we will talk about that today. The pigments in your skin actually protect you.
Oh yeah.
Actually protect you from UV radiation, and like we talked about last time, UV radiation is a known mutagen. It mutates your DNA, which increases your likelihood of cancer. It also breaks down your collagen and elastic fibers. So it makes your skin age as well. And so your skin, the color in your skin is actually trying to protect you from UV damage. Okay, so your skin also does both excretion and secretion. Secretion, again, you make things on purpose. You make some of your oils on purpose, right? And then you excrete some waste products on the surface of your skin. I don't know if you guys know this, but have you ever visited an old folks home in which people are on boatloads of medication? What's your skin smell like? It smells like medicine, doesn't it? Because you actually secrete those waste products or excrete those waste products onto the surface of your skin. If that doesn't make sense to you, then if you've ever gone on a tequila bender, the next day, what do you not want to smell but is coming out your damn pores? Tequila. Yes. It's like following you around, like your bad decisions. It's like, oh, did you really think that through, Melanie? I'm like, no, apparently I did not. And then your skin also protects you from temperature regulation 2 ways. First off, intact skin is like closing the doors in your house, so it's passive insulation. And of course, more with the subcutaneous layer, but we're just talking about the skin itself there too. And then in addition, your skin can get more active in temperature regulation by doing vasodilation to the surface when you're hot, and then you send your warm core blood to the cool surface, and then the air blows on it like soup, and it cools it down, and you can use that to cool the rest of your body. It's basically a little air conditioning thing, more like swamp cooler, right? Swamp cooler, yeah. So you got a swamp cooler thing going on, but also if you're really cold, you need to keep the warm blood close to the core, right? So you do vasoconstriction to the surface of your skin, because if you had to lose something, you want to lose a finger and a little bit of skin or a liver. Yeah, you want to lose a finger, right? So you do vasoconstriction. So it's actually helping to maintain your body temperature and homeostasis by doing this. Okay, your skin also does sensation. Really, really good place to do sensation because it's your boundary between you and the external world. So it's got touch, temperature, pressure, and pain sensation to let you know what's going on. Also does vitamin D synthesis. When exposed to little bits of UV radiation, you will make vitamin D in your skin. It has to be activated in your kidneys, but you make it in your skin. And of course, that's for calcium absorption, which is important for bones and other things. And then interestingly, your skin actually is a blood reservoir. What I mean is when you're sitting right here and you're not working out, you don't need all of your blood coursing through your skeletal muscles, right? But you need to be able to get it there at a moment's notice. So what happens is when you're inactive, the blood is just sort of slowly percolating through your skin, but then if you start working out, you vasoconstrict the vessels in your skin, send it back into circulation so that you can use it elsewhere. So it's just kind of this holding place for your blood. It's not sitting stationary, it's just going slowly through your skin. Okay, and specifically the dermis. It's also your skin is also temporary storage of a bunch of stuff: water, glucose, fats, and salts. And all of the women in here know that your skin can vary dramatically in the amount of water and salt that it stores, right? Now, that varies with what you eat. Of course, most of the men in here have experienced the fact that if you eat an entire 1-pound bag of Doritos, you will actually retain water the next day. You won't be able to get your rings off. Your pants may have seemed to have shrunk the size. But the women have experienced it more dramatically because under hormonal influence, you can take almost every damn bit of salt and fluid that you eat over the course of the day and you can all put it in your skin, right? And you're like, well, apparently I need to buy all new pants the next day and none of my rings fit, right? So the hormonal influence can just change the amount that you store in your skin. Okay, so now what we are going to do is we're going to start looking at the layers of the integument. We're going to do epidermis first, then we'll do dermis. Okay, so here we are. Here's the epidermis. You guys know what kind of tissue it's made out of, right? Okay, but if you look at it more closely, you can see that it's actually a little more complicated than it initially looks. So if you look at the epidermis more closely, you can see it's actually made of multiple layers called strata, singular is stratum, 4 to 5, depending on whether you're looking at thick skin or thin skin. And it's mostly keratinized stratified squamous epithelium. There's a few other kinds of cells in there. And remember, of course, even though it's called keratinized stratified squamous epithelium, the cells at the bottom don't look squamous, right? Just the whole tissue is called squamous because of the shape of the cells at the apical surface, right? Okay, so what we're going to do is we're going to build the epidermis the way that it actually grows in you. And the way that it grows in you is deep to superficial. Yeah? So the newest cells are here, the oldest cells are there, and on average, depending on your age, you get a complete turnover of your epidermis about every 30 days, right? So new ones go all the way up to the top and you slough them off. It's continuous, right? It's continuous, making new cells and sloughing off old ones. So how the hell can you ever have permanent skin damage? If you have a scar, that means it didn't actually just damage your epidermis, it damaged your dermis, which does not have a turnover rate of every 30 days. That makes sense? Okay, so what we're going to do is we're going to build the epidermis and we're going to start at the bottom layer. There's a bunch going on the bottom layer. Okay, first let's look at the epidermis in 2 different kinds of skin. So this is typical skin, probably thick skin, but let's look at the difference in thickness between thin and thick skin. Here is thick skin going from here all the way to the surface. That maybe is the palm of the hand or the sole of the foot or something. That's pretty thick. And then here is thick skin going from here to the surface. That maybe is around the lips or around the eyes. Does that make sense? So it varies pretty dramatically, but it has mostly the same structures once you look into it, the thin skin versus the thick skin. Okay, so the bottom layer down here is stuck to the basement membrane. It's at the basal surface, so it's called the stratum basale, the basal layer, which is what stratum basale means, okay? This layer is really, really active in cell division. It goes through cell division relatively rapidly. And one of the things that's cool about this layer is that not only are these guys really active in cell division, but they're all sort of formed as a cohort or a team. So when they are made, they are all attached to one another laterally by desmosomes. And then they move up together. Now, some of them go through cell division faster than others, so it's not like they're just going to stay one single layer. But the important thing is, since they all move up together, that means that, and they all have desmosomes, that means that if my skin is really dry, it probably is right now, when I do this, you can actually see the skin flaking off because it doesn't flake off as individual cells, it flakes off as big groups that were stuck together, right, with desmosomes, right? So if that doesn't make sense, think about this. What if I get sunburn, right, and I damage all of them at the same time? Then give me a few days, and by the time they have moved to the surface, almost none of the desmosomes have broken, and then they just all pull off as one big sheet, right? If you give them a little bit more time, then more of the desmosomes are going to break. Does that make sense? Okay, so this bottom layer is the stratum basale. You can even see some of the cells going through cell division down there. And it's attached to the dermis by a basement membrane, which is like the sticky stuff, plus some hemidesmosomes. And it's a single layer of cells that look more columnar than anything else. They're usually columnar or kind of cuboidal, but you don't call them that. They just look like that. And these cells down at this layer are called either basal cells, obvious, right? Or keratinocytes. Not because they're producing keratin yet, but they will eventually produce keratin, okay? And then there's some other cells in there, which we'll learn in just a second. So what is going to happen as you go through cell division and exfoliation is this. Cells move up, they're exfoliated off the top. Cells move up, they're exfoliated off the top. As you age, one of those two processes will slow and the other one won't. Cell division or exfoliation? Cell division will slow, but exfoliation is a mechanical process, so it doesn't slow, right? So what tends to happen to your skin's thickness as you age? It thins. And not just the epidermis, that's what we're talking about now. The dermis will also thin because you're going to start to lose lose collagen and elastic fibers. Does that make sense? So the skin will thin, and we probably talked about this before, but this is really, really important if you're dealing with elderly populations. You actually have to be really careful with skin when you're moving elderly people, or else you can actually tear their skin. If you get somebody in their 80s or 90s, you can move them improperly and make a rip right through their skin. So this bottom layer of basal cells or keratinocytes are eventually going to start producing keratin. They don't start producing it until they get up here, but they will soon. And what's going to happen with these guys as they start to produce keratin is by the time it gets to the top, the cells are going to be dead, and you'll just have basically a bag filled with keratin that's going to stay around. Okay? That layer of keratin, you are going to shed, move it up to the top and shed it, again, about every 30 days. You end up shedding about 18 pounds of skin in your lifetime. Most of it's in your house as dust. So just make sure you're living with people that you wouldn't mind snorting up some of their cells, because you're totally doing it, right? Which is super gross considering how many teenagers are in my house at any given time. And also, 11-year-old boys, why the hell can they not pee all over everything? I would like to know about that. I started making my son's friends give me $1 every time I sit and pee. I'm like, give me $1. He's like, I don't have $1. I'm like, here's $5. If you can get through today without me sitting in pee, I will give it to you. Never once have they ever. Okay, so what is dandruff related to this story? Dandruff is when you are super duper duper effective at producing keratin and then sloughing off the keratin. okay? You don't just do it on your head, you do it everywhere, but it shows on your head, especially if you have dark hair and wear dark clothes, right? So it doesn't have anything to do with hygiene, although if you have poor hygiene, it'll start to accumulate, right? And you'll see it, right? So a person with dandruff has just a really fast rate of keratinization and therefore exfoliation. You're going to see it, right? So how do like the over-the-counter dandruff shampoos work? When your hair is wet, it's just really, really exfoliating, but when it dries, it just sticks to your head until the next time, right? That's what it does, right? It just doesn't let anything out once it's dry. Now there are prescription medications that actually do things that are slightly different, but like Head and Shoulders, that's how that works. Okay. All right. So now let's talk about the first kind of cancer. So I'll teach you the three biggies for skin cancer. There are more skin cancers than this, but the three biggies. The first one is called basal cell carcinoma, and it occurs in these cells, which have a really rapid rate of cell division anyway. So don't be at all surprised that it is a very common type of skin cancer. Unfortunately, your textbook only shows it in the late stages. That is not helpful to anybody, because if you've got like a big wet booger that just stays on your skin, you'll know something's wrong. But what about in the early stages in which it looks like this? It doesn't really look quite as concerning, especially if it were smaller than that. Usually it's a shiny dome. Sometimes it's red when it gets a lot of capillaries into it, but sometimes it's just flesh colored. So it might look like a zit that just never goes away, right? And it's really, really slow to metastasize compared to the other two types of skin cancers we're going to learn. This one is called basal cell carcinoma. It's the most common, least likely to metastasize. Can it kill you? Of course it can. Any cancer can kill you. But usually it has a full cure by surgical excision without even radiation or chemotherapy, as long as you catch it early enough before it's metastasized. About 99% of the time, people are going to get over it with this one. But can it kill you? Of course it can, because it's cancer. Let's talk about where people get cancer. It's kind of sex-specific, but it's also cultural. Because the place in which you're more likely to get skin cancer is wherever you are exposed to UV radiation, right? And so, of course, this is dramatically different here than it is in Afghanistan, right? Because I ain't going to have much exposed at all in Afghanistan. However, it's still different in males and females here. So let's talk about the United States. So in the United States, any idea where women are more likely to get skin cancers? Any idea? Usually your chest. Why not your face? Because most of us at some point get vain enough that either we wear makeup or we wear skincare products to protect our skin, right? So let's do a poll. How many women in here wear something on their face that has sunscreen in it? Okay, now let's do the other poll. Men. How many men in here wear something in their face that has sunscreen in it? All right, now guess where men get skin cancer? On their face. Yes, they do. They get it on their face. Okay, so women get skin cancer more likely here and on their hands. Guys, if I could go back and redo my 20s, this is one of the things that I would redo. I would wear more protection here and more protection here. Women also, although not so much in Utah, get skin cancer on the backs of their legs. Why is that? Because we wear shorter stuff, and after a certain age, you don't even want to look back there. You're like, I don't even know what's going on, but I'm sure it's fine, right? It'll be like 2 years before you look and things like that big. You're like, I don't know, it's fine when I was 18, last time I looked back there. And then men, in addition to their face, backs of their ears, back of their neck, as you start to lose your hair, get it on your head, on your hands. That's where men tend to get skin cancers. If we could convince our loved men that they should wear sunscreen, they wouldn't get it as much on their face. But good luck with that. Okay, so that's basal cell carcinoma. What else is down there in that level? Oh, by the way, as basal cell gets worse, it starts to look entirely different. What else is down there on that level is your first sensation. The most superficial sensation that you have is actually in the deepest layer of your epidermis. And that's these cells right here that are attached to the sensory nerve ending. These are called tactile cells or Merkel cells. That means that the entire other layers, the layers superficial of that, have no sensation. And then remember that the entire epidermis also doesn't have any blood supply, right? So did anybody else do this, or was it just redneck kids like me? Did you guys torture your siblings by sticking straight pins in your fingers and then running around after them? Did you? Why did you not do this? I guess it's because I didn't have any toys. I had like a stick and some straight pins. So what you did is you stick them in there. And why the hell doesn't it hurt? Well, because you don't have any sensory nerve endings. And you can tell when you hit the dermis because then it'll start to bleed, right? And the idea was to terrorize them, not you, right? So you can totally stick them in there and it doesn't even hurt at all until you get all the way down to the deepest layers. So those are tactile cells. Now the dermis has boatloads of sensation. If you hit that, you're gonna know. Oh, here's a reference that even non-redneck people will understand. If you get a blister, what happens is you irritate the epidermis and the dermis enough that they separate, and you pull the tactile cells away from the sensory nerve endings and build up fluid in there. Do you feel anything on the surface? Nope. But if you take the blister off, do you feel anything? Oh, hell yeah. Because that's actually the surface of the dermis you're coming in contact with, and that's where all the sensory nerve endings are. Okay, so what else is down here? The other stuff that's down there is melanocytes. And melanocytes, do you see this little guy right here? Have you guys seen Nightmare Before Christmas? He looks like Oogie Boogie to me. He does, right? Oogie Boogie. Okay, my kids are terrorized by Nightmare Before Christmas. I think it's so good and they hate it. I do too. Those are melanocytes. We're going to talk about them in more detail in just a second. They are responsible in part for the color of your skin, but skin is not just one color. There are at least 3, arguably more, pigments that go into making your skin whatever color it is. And we know, of course, that skin is not black or white, right? That's not really how skin works. There's like an incredible range of variation. That's not even all of it, of course. You can get far fairer, far darker. But in addition, before we get to melanocyte, let's talk about the other 2 pigments that give your skin its color and its tone, right? So melanocytes are responsible for the brown, yellow, brown, or black pigments, okay? And then the other 2 tones in your skin are because of how much hemoglobin is in there. So if you have capillaries really close to the surface, then your skin's gonna have that kind of that rosy or ruddy tint to it, okay? And then the other tone is because of keratine, and keratine is a yellowish or greenish pigment. And those of us who have olive skin, I have olive skin, you have olive skin. It can be olive light or olive dark. It just means it's got that yellow tone underneath it, right? And probably if you have olive skin, when you start to get sick, you look like hell. Like you're just like yellowy green and people want to save you. You're like, what the hell is going on here? I'm like, oh no, I just got a cold. So that's olive. So that's keratine. But then in addition to the keratine and the hemoglobin, you also have melanin. So melanin is a really interesting pigment. It's especially interesting because of its cultural significance, and we'll talk about that some more next time. But let's start talking about melanin today. Okay, so your melanocytes, those oogie boogies, are going to produce melanin. So I want to talk about the difference in melanocytes and skin color. We start talking about skin color. What I'm talking about is your inherent skin color, not what happens when you tan. We'll talk about tanning later, right? So whatever skin color you have, right, if you haven't been tanning or it's the middle of winter, right? So what is the difference between people with inherently dark skin and inherently light skin? It's not the number of melanocytes, because we all have pretty even number of melanocytes. I mean, there's some variation, but it's not based on population. What is really different with people with inherently dark skin is their melanin production from their melanocytes is much higher. They have much higher protein synthesis. And the melanin that they make persists in their skin longer. Yes? And it also goes more superficial, so it's more protective. Does that make sense to you? So let's look at what melanin does. Okay. Melanin is a brown, yellow, brown, or black pigment. The color of it is basically related to your genetics. There's variations in the color that your melanin is. Okay? So, in a person with inherently dark skin, what happens is these melanocytes, your oogie boogie, is going to make little vesicles filled with melanin. Yes, it does that with all of us. And what it does is it makes them, and some of them stay inside, but notice some of them actually leave. Yeah? In a person with inherently dark skin, the melanocytes are not all at the bottom. Some of them go into the more superficial layers. And when you make melanin, it actually goes and spreads out more and stays around longer. But what happens in all of us is the site at which your melanin is deposited is wherever you get hit with UV radiation. So you get hit with UV radiation, the melanin will deposit on that side. What's the job of melanin? The job of melanin is to protect your cells, specifically to protect your DNA. Because UV radiation is a known mutagen. It mutates your DNA. It also can cause cancer. And the function of melanin is like putting on a dark shirt in the summer, right? To basically bounce off the UV radiation so that it actually doesn't do damage. So now I want to talk about people with inherently darker skin versus inherently lighter skin and risks of cancer and also skin aging. Okay, so if you have somebody with inherently darker skin, someone who's from sub-Saharan Africa or their family's from sub-Saharan Africa, what do you predict about their likelihood of skin cancer compared to, for instance, me? Much lower. Now, am I saying people from Africa never get skin cancer? No, because cancer's dick. You can get cancer anywhere for any reason. It just does what it wants. But if you're talking about statistical likelihood, I have a much greater likelihood, right? And you, with lovely fair skin, probably some Irish or Scottish, have very little protection, comparatively speaking, to UV radiation. So I really hope that you wear sunscreen, and most of us do. Now, what about aging? If UV radiation also breaks down your collagen and elastic fibers, what can you tell me about how the skin is going to age in someone with inherently dark skin from, for instance, sub-Saharan Africa and someone from Ireland? How is skin aging going to go? Who's going to age better? Oh, the person with dark skin. I don't know if you have ever seen how incredibly dramatic it is how well people with dark skin age. Like, they can look 30 years younger than someone with light skin. And it's because of what you have with UV radiation is simply that you are progressively breaking down your collagen fibers. And it doesn't really show at all when you're young because your rate of replenishment of collagen fibers is totally fine. But later on, after you can't replenish your collagen fibers, you show the damage of every single bit of breakdown of collagen fibers. Does that make sense? Okay, so these are your melanocytes. So melanin is responsible in part, but not completely, for the color of your skin, your hair, and your eyes. Okay, so you can have a lot of melanin deposit in your skin, but not very much in your hair. You can have it in both places. Those of you who have blue eyes, you have very little melanin deposit on the surface of your eyes. Do you have any idea what eye color has the most likelihood of UV damage? Blue. If you have blue eyes, you need some good sunglasses because you have over time more likelihood of UV damage in your eyes. So invest in some good sunglasses, especially as you age, and that UV damage can eventually cause vision loss as well. Okay, so skin color variation. We can look at a little bit of skin color variation here and talk about why it occurs. But before we get to the skin color variation and why it occurs, let's talk about a couple of disorders in which you can't produce adequate amounts of UV. Hold on, I forgot to pull these up, so give me a second. Yeah, we're going to do vitiligo. Oh, who's this beautiful, beautiful woman with vitiligo? Oh, there she is. Can't remember her name. Her name is Winnie something. So vitiligo doesn't usually look like this. She's absolutely amazing, skin color or not. So what vitiligo is, it's an autoimmune disorder in which your immune system makes a mistake and starts to destroy your melanocytes. And when you start to destroy your melanocytes, you can't produce any pigment. And of course, it's going to have a relatively dramatic effect if your skin is inherently dark, right? But people with inherently light skin can also get vitiligo. So let me show you some other vitiligo. This is autoimmune, so there is no cure for it. This is a little girl with vitiligo.
So will it usually take place when the skin is thinner?
No, it's just random where it hits. For some reason, it seems to hit bilaterally. We don't know why. And sometimes relatively symmetrically, but there's no cure for it, and sometimes it goes into remission, and sometimes it comes back. So you can see the difference. This person has inherently dark skin, right, and this person has inherently lighter skin, but they've both got vitiligo. Now, the areas that are not producing melanin are at much greater risk for UV damage and also for skin cancer, so you have to protect your skin if you've got vitiligo. This is what Michael Jackson said that he had, which is how his skin got light, and he may very well have had it in the beginning. But that's not what happened to the rest of his skin. So it does not do an even bleaching all over the skin. So vitiligo may have been the initial stages, but then some other stuff happened, probably involving lots and lots of money. So that's vitiligo. And then let's talk about albinism. Albinism is not autoimmune, it's genetic. So let me find you albinism for a second. Okay. This is a little boy of African descent with albinism. He doesn't have complete albinism of the eyes. Let me find you guys a couple other pictures. This is a person not of African descent. I don't know what, but they have albinism. Okay, so what albinism is basically there's a whole biochemical pathway to produce melanin, and there are several different genetic mutations that can occur in that biochemical pathway. And if something occurs in the biochemical pathway and you can't make the final product, then you're not going to produce any melanin. Now, it can occur in the hair, the skin, the eyes, or all three, okay? And when it occurs, of course, this child has almost no protection from UV radiation. So both the hair, the skin, and the eyes are all completely susceptible to UV damage. Kids with albinism to this area, to this level, especially with the eyes, are going to be likely to have UV damage in the eyes. They usually have blindness as one of the risks, like a progressive degeneration of your sight as one of the risks. So why do the eyes show up pink, whether it's an albino human or some kind of other albino mammal? Any idea? What?
You're actually seeing like the retina.
Yes, exactly. You're seeing all of the vascular structures inside the eye, which would normally be protected with 2 layers of pigment, right? Those of us with darker eyes, does anyone have really dark eyes? Really dark eyes, what you have is you have a blue layer in the back that everybody has unless they've got albinism. And then you have another layer on top, and those are 2 layers that are protecting all of the deeper structures. If you have really light blue eyes, then what you have is just a blue layer behind and nothing on the top. These guys have none of those pigments. Right? And how do you get like green eyes? Well, what you get is you get the blue layer in the back and then some yellow pigments on the top, right? So it's like the layering that goes on in there. So this is albinism of someone, and this is albinism in which he doesn't have complete albinism of his eyes as well. You see that? Okay, so albinism is generally genetic. It can just be because of a mutation, but it increases in likelihood with inbreeding because most forms of albinism are autosomal recessive. What do I mean by that? You need a double dose of the mutation, yes? One from mom and one from dad. Relatively unlikely to get 2 of the same mutated allele unless your parents are cousins. And then you have increased the likelihood. Now that sounds bizarre to us now, but historically speaking, it has not been at all uncommon. Especially since, historically speaking, it was one way to keep the money in the family, was it not? You just marry in the family. Have it left, right, and center with royals, right? Entire history of European royalty. You guys know that Cleopatra was married to her brother. Super weird. Now, albinism has become, have you guys seen albinism in the news here lately? Because there are some areas of Africa in which albino people, and specifically children, are really, really at high risk because the parts of albino children are considered really, really lucky and part of some rituals. And so if you have albino children, they can, for instance, get attacked and get their limbs cut off and those kinds of things. So a lot of times they're actually removing. Look at that, it's really fascinating. I can't remember which country it's in. So anyway, that has been happening, and that's happening up to this day. So the function of melanin, and we'll talk about skin color variations. Next time we'll talk about tanning too. The function of melanin is really to shield the epidermis and the dermis from UV radiation. And the more melanin you have, the better protection that you have. But that's not true when you tan, because what is really happening, guys, when you tan is that you are blasting with UV radiation. And what you're trying to do is you're trying to keep up with it. You're trying to keep up with what's happening with your UV radiation. I love this cute little picture. What's wrong, melanocyte? She's tanning again. He's trying his best to keep up with it, right? To keep up with it. And usually if you lose control and you haven't keeping up with it, then you'll get a sunburn. If you have a sunburn, you know that you have actually allowed the UV radiation to damage the tissues. It doesn't necessarily mean that you're going to get cancer, but it increases your stats really dramatically. And we'll talk about burning in children and stuff in the next class. Okay, I'll see you guys. Unless you're coming to my office, I'll see you Thursday. Remember, I don't have office hours this Thursday. Oh, can I have the short answer part back, please? Oh, sorry, I should have gotten all that stuff out of your space before now. And you can take notes on this when you come to my office too. You can keep the Scantron. You'll need it to check your answers. Thank you, thank you, thank you, thank you. Thank you. It's somewhere around here.
Okay, can I just look through it?
Yeah,
or-
but you can't keep it. You can look at the short answer and you can look at the other part of my office.
Thanks, Molly.
Thank you. You guys did much better.
Yes, you did.
Yes.
Great. Yeah.
Did you go talk to MJ or no?
No, because I work Tuesdays and Thursdays, and then when did I go? I went on Monday.
Don't even bother this week. She's gone for a week. You can send her an email. I don't know if she'll answer it, but she can send it. I want to tell her that you made an A so that she'll be more likely to let you in. But if it doesn't happen, I've got one other possibility for you, but I don't